Hello friends, this video on Biomolecules Part 9 is brought to you by examfear.com. No more fear from exam. So that was all about disaccharides. Let us now talk about polysaccharides. So polysaccharides are those which on hydrolysis give multiple units of monosaccharides. So they contain a large number of monosaccharide units joined together by glycosidic linkages. So there are too many glycosidic linkages. So here you can see this is one glycosidic linkage, this is another glycosidic linkage. Again here there will be another glycosidic linkage and so on. So there will be multiple glycosidic linkages connecting multiple monosaccharide units. So that is polysaccharides. These are most commonly encountered carbohydrates in nature in the complex forms, because when you talk about polysaccharides, the examples are starch, glycogen, cellulose. So these are the things which are very commonly present everywhere, because when you talk about starch and cellulose, they are abundantly found in plants, and you know how many plants are there. There are plants everywhere. So these polysaccharides are very much abundant in nature. They mainly act as the food storage or structural materials. So when you talk about food storage, we talk about starch in case of plants and glycogen in case of animals. I mentioned this before also, why they act as energy storage, because they are made up of many monosaccharides unit. So if that energy needs to be utilized instantly, that is little difficult, because the monosaccharides will get hydrolyzed to form the polysaccharides will get hydrolyzed to form monosaccharides, and then those monosaccharides can be instant source of energy. So that ways it cannot be instant source of energy. So that is why they act as energy reserves or energy storage. They are also used as structural materials. What do we mean by structural materials? For example, cellulose. Cellulose is the it makes up structures, or it builds structure. So cellulose is something which is seen in the cell wall of plant cells. So every plant at the end of the day is made up of plant cells, and the plant cells have cell walls, and the cell walls are made up of cellulose. So that way, cellulose is a very important structural material. Similarly, another example would be chitin. So chitin is another structural material which is generally seen in the exoskeleton of cells. So that way, polysaccharides are used as both food storage as well as structural material. Examples would be starch, cellulose, glycogen. So the three most common examples of polysaccharides. So let us talk about starch. So starch is a polymer of glucose. So several glucose units together form starch. Now there are two forms of starch which are generally seen, amylose and amylopectin. So let us see what is amylose and what is amylopectin. Now before that, where do we find starch? Starch is found in cereals. It is also found in vegetables. When I talk about cereals, one common place where you would have often seen starch is rice. When you prepare rice, you would have seen your moms preparing rice in a vessel, and then a watery, sticky, viscous substance comes out of that rice. So that is nothing but starch, and that is often used to put it in your clothes to make them stiff before you can iron them, especially on the cotton clothes. So when you wash your cotton clothes, they generally become very soft and they are not stiff. So in order to make them stiff, so that when you iron them or when you press them, they look really bright and they have that proper shape. So for that, you often starch your clothes. So that starch is nothing but this polysaccharide starch, which is found in abundance in most of the cereals like rice. Now there are two forms in which starch can exist. One is amylose and the other is amylopectin. So let us see each of them. So when I talk about amylose is water soluble, and it is a linear structure, somewhat like this, as you can see here, a structure of this form. So here glucose units are joined together linearly by glycosidic linkages. So that is how they are connected. So it forms a linear chain-like structure. So that is amylose. So this amylose form of starch is water soluble. That is also one thing to remember. So amylose form is water soluble, whereas amylopectin is water insoluble. In amylopectin, the structure is branched. So here again we have the glycosidic linkages. If you see here, you have glycosidic linkages between C1, C4 like this, but you also have glycosidic linkages between C1 and C6, that is the branches. So here you can see this is like linear structure. So this structure is formed by C1, C4 linkage. Again, this structure is formed by C1, C4 linkage. This is formed by C1, C4 linkage. But wherever you see branching, like here, 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 so wherever you observe branching, all these branching is because of this linkage, the linkage between C1 and C6. So this is also a glycosidic bond, that is, this is also a COC bond. This bond also is formed by loss of a water molecule. So this type of bond which exists between C1 and C6, that gives rise to branches. Otherwise, they are also similar to amylose. So if you consider only this part of the structure, this is similar to amylose. This part of the structure is also similar to amylose, but they are interconnected by these branching bonds, that is some additional glycosidic linkage. So here you have C1, C4 linkage and C1, C6 linkage. But here you have only C1, C4 linkage. So that is why this is linear and this is branched. This is water soluble, whereas this is water insoluble. So these are the two forms of starch, amylose and amylopectin. Now when you talk about some of the applications of starch where it is used, it is generally used as an adhesive. As I said, it is also used for stiffening clothes. It is also used for thickening in cooking. For example, in thickening soups and all, there also it is used. And appearance wise, it is white, tasteless, odorless, and it is generally insoluble in cold water. However, it is soluble in warm water. Let us talk about the next polysaccharide, that is cellulose. So cellulose is a straight chain polysaccharide composed of glucose units which are joined by glycosidic linkage between C1 and C4 of the next glucose unit. So this is very similar to the structure of amylose. So here also you have C1, C4 linkage. Here also you have only glucose units. But as I mentioned before also, I was telling you, right, that starch and cellulose, they are mostly the same, but still the arrangement is different. Like where you have carbon, where you have hydrogen, where you have oxygen, that placement is different. That is why the arrangement is different, and that makes a huge difference in their properties. So when you compare cellulose and amylose, they are very similar. Both of them have glucose units, both of them are straight chain, both of them have C1, C4 glycosidic linkage. But still there is a lot of difference in their properties. So if you ask where are they are found in the cell walls of plant cells, and it is exclusively present in plant cells because we don't have cell walls in animal cells. So here you can see the glycosidic linkages which are like this, a linear linkage. Now the third polysaccharide, that is glycogen. The way starch is used for storing food in case of plants, similarly in animals the food is stored in the form of glycogen. So when is this energy utilized? For example, however, in animals the primary storage of energy is fats. So fats are the primary storage of energy. However, when you have too much of fats, that might lead to obesity and that might give you health issues. But the primary storage of energy is fat. The secondary storage of energy in animals is glycogen. So the structure of glycogen is very similar to amylopectin. So you can remember it this way. Glycogen is similar to amylopectin. So both of these are branched. And amylose is similar to cellulose, and both of them are linear chain. So this is a multi-branched polysaccharide that serves as a form of energy storage in animals and some fungi as well. So this is how the structure looks like. You have C1, C4 glycosidic linkage this way, and you also have C1C6 glycosidic linkage this way. So these are the branches and these are the linear chains. So this is how the structure of glycogen looks like. So you can see it is very similar to the structure of amylopectin. So you have the straight chains here, that is the C1C4 linkages. So these are the straight chains. Now wherever you see branches, so these points are the C1C6 linkages. So these two types of linkages are present even in case of glycogen. So let us have a quick comparison of the three polysaccharides which we discussed just now: starch, cellulose and glycogen. So if you look at all of their structure, there is one similarity, that is all of them are made up of multiple glucose units. But if you talk about starch, they are available in two forms, amylose and amylopectin. So one form is a straight chain where you only have C1C4 linkages, the other form is branched one. Whereas in case of cellulose, it is only a straight chain similar to amylose, and in case of glycogen, it is similar to amylopectin with branched chains, that is C1C4 linkages as well as C1C6 linkages. So now, are you getting an idea that how different monosaccharide units can get linked to form polysaccharide? So the same monosaccharide unit, that is the same glucose, is getting connected in many different ways to form different polysaccharides which have properties so different from each other, like starch, cellulose and glycogen. Now as I mentioned before also that starch is the As I mentioned before, that since starch is used for storage in plants, similarly glycogen is used for storage in animals. That is why glycogen is often known as animal starch, because it serves the same role in animals as the starch does in plants. So I think with this we have discussed almost everything about the carbohydrate. So let us quickly discuss the significance of carbohydrates in life. They form a major portion of our food. For example, the vegetables, the cereals which we eat, they all are a good source of carbohydrates. Carbohydrates are used as storage molecules as starch in plants and glycogen in animals. So however, in animals, glycogen is the secondary storage of energy. So initially the primary storage is fat. So whenever the body needs some energy, so the first help it takes from fats. Now if not enough fats are there in the body, then glycogen is utilized. This glycogen is generally stored in liver and muscles of animals. Cell wall of bacteria and plants is made up of cellulose, so it acts as a structural material. It is also used in furnitures, paper, cotton textiles. So these are again some of the commercial uses of carbohydrates, as well as medicines. So with this we end our discussion on carbohydrates. Now also breweries, for example, the maltose sugar which we spoke about, the disaccharide which is made up of 2 glucose units, it is also helpful in baking or making beer. So in breweries also it is helpful. So with this we end our discussion on carbohydrates. So we have discussed all the types of carbohydrates, how they are prepared, how they are useful, what is their structure. So we have discussed the most of it. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.